Welcome to Thursday Dairy Update. This is Kathy Barrett with the Cornell PRODAIRY program, and along with Dairy Business Communications, we're pleased to offer this webinar series. Today we have Dr. Heather Huson, who is a professor at Cornell, animal geneticist, and she will be talking about genetics on the farm.
We're going to start with my presentation, and really what this is looking at genetics on the farm. And for the most part, what I'm going to focus on today is genetic evaluations. So just to put a little bit of perspective though, all of my research, the majority of my research is on dairy cattle genetics. And the pictures that you see here on this slide are all students of mine, graduate and undergraduate students. Our main projects right now are looking at the genetics of mastitis, of lameness, and ketosis in dairy cattle. So that's some of the research that's going on in my own lab. The presentation, what I'll be going over is basically what are genetic evaluations, and then how do genetic evaluations basically get from the farm, get processed, and then back to the farm, what's included in those evaluations, how to use the information that you get back, and lastly, who would you actually genotype on your farm? So the first is, what are genetic evaluations? So the main point of this is that it's an estimation of genetic merit of your animal. So commonly what you'll hear is genotypic value, and so that's an individual's genes based on their own performance. So what do those individual genes of an animal, how do they influence that animal's performance? Breeding value and progeny difference are both how the animal's genes affect their progeny's performance, or what does the progeny inherit. So most often you'll hear EBVs, which is another term for estimated breeding value. Often if you see a small g in front of that, that's talking about genomic EBVs. Similarly, you'll have PTA for transmitting ability, and GPTA is genomic transmitting ability. So traditional genomic evaluations have always been done really based on the cow's phenotype. So all of our economically important traits, their production, their health, conformation, as well as using pedigree information. So putting the two of those factors together, and pedigree is really used to base on, if it's a bull and we want to give a merit on milk production, the bull isn't going to have milk production. So we're using his pedigree to look at his daughters and what's a daughter's milk production. We put those statistics together with the phenotypes and we have a predicted genetic merit. So those are traditional evaluations. Now what we do is we add genotype. So genotype is the DNA of the animal. So what that adds is basically, this is your single slide that you need to deal with biology here. So what the genetics is, what's that based upon, is really what we call marker-assisted selection. So SNP is a single nucleotide polymorphism. So really what we're doing is we're comparing these SNPs in your general population compared to your affected population. So affected in this case could be the really high-performing milk producers. So for instance, the frequency of those SNPs. So in the general population, you might have a SNP that carries the allele G as opposed to a SNP that carries the allele T in the really high-producing animals. So we take basically those statistics and we associate them in a genome-wide association study where we associate the SNP with the performance or health trait that we're looking at. So that's how this genotype comes into play. It's basically these markers tell us that this animal is more likely to be high-producing or resistant to a particular disease as opposed to the general population. So what does this actually give us for a benefit? So what you can see in this particular chart is looking at protein reliability. So using traditional evaluation, so that's based on what do we actually see in the animals. So even though this is looking at bulls, what they did is take the protein production of daughters, and they can estimate using traditional evaluations with about a 40% reliability, that's the blue peak, of how well that animal produces protein, what the protein reliability is. So if you're selecting for protein content in your animals, there's about a 40% reliability when selecting for that protein content. Now if you add the genetics, so that marker-assisted selection, your reliability goes from about 40% in selection to about 75% for selection. So the genomics is really adding increased accuracy and reliability for selecting for that trait. So how do we use this information? So basically that phenotype and pedigree are often, this is kind of seen as our proven genetic evaluations. And this works. It works well. The reliability is a little bit less, but it is proven, and some people are more confident in these evaluations. One of the drawbacks is that they're very timely and costly to do. So if you want to know, a genetic evaluation on a bull, you generally have to wait almost 5 years by the time the bull is born, till it is old enough to breed, and then till it has daughters that are actually producing to create a genetic evaluation. So that's quite a bit of time and quite a bit of money. The genomics, like I said, it increases your accuracy, and it's particularly relevant if you want to select on young stock. So instead of waiting until you can get that proven information, what is the animal's potential as a heifer or a calf, or a young bull? Bottom line is, when you can put both the phenotypic and the genomic information together, that is the most reliable genetic evaluation for your animal. And in the long scheme of things, what you're going to get is basically you can have young genomic evaluations. As the animal ages, it will start adding the phenotypic information that comes into the council, and you will have a combination of both genomic and phenotypic genetic evaluations. So how do we actually get these on the farm? So generally the first line of contact to get basically from the farm to get a genotype is usually through industry, and this can be like the genotyping industries. And I put a couple logos on here. These are not all of the places that you can go to. It's just a few, and it's the one that I most commonly work with. So usually you'll get genetic evaluations through genotyping industry, your breed associations, and the other main point of contact is the Council on Dairy Cattle Breeding, which I'll talk about in just a minute. But for instance, Zoetis with their Enlighten CLARIFIDE programs are one genetic evaluation. GeneSeek is another one that provides genetic evaluations. And then a lot of these are also done through the breed organizations such as HOLSTEIN and Jersey. So to mention the Council on Dairy Cattle Breeding, this is who actually generates the genetic evaluation, so the scores that you would receive. So this used to be performed by the USDA, and much of the foundational research was done by the USDA and is still done by the USDA, also bringing in individual research like myself at universities. They will also incorporate that into genetic evaluations. But the CDCB has a number of individuals here that are represented in their group. So this isn't just a single group, even just academically, that comes in and determines how we perform genetic evaluations. So there's 3 members from each of the above organizations that are included in the CDCB. There's a total of 12 voting members, and there's also 2 non-voting industry members. So this is the main organization, and they are a good point of contact if you have any major questions about what's included in your genetic evaluations, and they have a huge website. I will admit it is not the most user-friendly website, but there is tons of information on how genetic evaluations have evolved, what's included, even the economic values. So in the big scheme of things, when it comes to genetic valuations, we rely on dairy herd information. So DHI is basically contributing a lot of that herd information, production information. We work with AI organizations and breed associations, and also the DNA laboratories that process this. And it's exchange among all of those to and from the Council on Dairy Cattle Breeding. So unfortunately, when you decide you want to have genomic testing, it's not really as straightforward as we would hope. So even though I mentioned just a couple of these different groups, just within Zoetis, you can have the CLARIFIDE panel, but there's a low density and a high density panel. Within GeneSeek, so that's Neogen, there's multiple. I only listed 3 of their panels. They have multiple panels available. You can look at the HOLSTEIN website, and there's the HOLSTEIN Complete, which has a number of individual tests as well. Plus, the CDCB will also have different fees associated with their council. So to put some of this in perspective, some of these tests are individual tests. So such as under the HOLSTEIN Complete, they are individual tests for, say, looking at polled or horned. That's $40. Whereas a lot of these programs now, you will get kind of this compiled data back of individual tests as well as the genetic evaluations. That's what you most commonly see. But keep it all in perspective. So some of the things when you're looking at these different tests, to kind of help you decide which one there is, the more SNPs, so the more markers. So for instance, when you look under Zoetis CLARIFIDE, they have a low density panel that's 19,000 SNPs. They have a high density panel that's 60,000 SNPs. There's a huge price difference there. What that means is generally, the more SNPs, the higher accuracy of predicting that particular trait. So when we were talking about that protein reliability, it would increase the accuracy of predicting that trait. However, often we have this down packed pretty well so that your prediction is generally a difference of, are you predicting it at an accuracy of, say, 90% for the low density versus, say, 98% for the high density? So economically, is it worth you spending more money on high density when you're still predicting at 90% for the low density? So that's something for you to consider. So with that in mind, my personal opinion, what I recommend is for most people, if they want genetic evaluations on their herd, for the most part, low density is just fine. The reason you might use high density is if you really have a particular bull that you're marketing, or maybe even a really high-end cow that you want to market. Those might be situations where the high density might be the appropriate choice. But for the average user, just selecting your animals, low density is normally going to serve the purpose. So an example of what some of these submission forms look like. So this is an example of the Zoetis CLARIFIDE. So the type of information you would have to put in here, this is actually their submission sheet, and it comes on basically an Excel file. And so you have your sample ID, your farm designation, your breed. Most of this is very basic information that you would all have on hand. You do have the option for Holsteins that they might be registered, but you can also do non-registered animals here as well. And you put in, to the best of your knowledge, what the sire and dam information is. So that's what a Zoetis submission looks like. For GeneSeq, this is fairly similar. This is one of their lowest panels, density panels. So they're about $25. If you were able to blow up the screen, the file on the right-hand side, though, what you can see is the original panel is $25. Now, if you want a bunch of the other individual tests, you immediately start putting on another $10, $20. So that can really add up. So sometimes beware when you're choosing the panel that you are getting the panel that has the items that you're interested in. But the information that you submit is very similar with farm ID, animal ID, sire and dam, and breed. So that's all very similar. So how do we actually get the DNA? So there's a number of ways that you can submit a sample for DNA. So most commonly on the farm, you're going to use hair samples. And these are pretty easy. The biggest thing to keep in mind is that you need the bulb from the hair follicle. So you can't shave an animal and send in a hair sample. You can, on the other hand, pull hair from pretty much anywhere on the animal. Tail and is one of the easiest places for the cows because it's longer hairs and easier for you to grasp and pull out. As long as they have the hair bulb, you're just fine. Blood from the tail vein or the neck vein are most common. And often some of these suppliers will provide you with these cards that you can put the blood onto. A tube of blood that your veterinarian collects as well or that you collect is also completely fine. The other one that's really common on farm, and the company that tends to do a lot of this is Allflex, but they're not the only company, but is taking a tissue sample, particularly ear punches. So these are the exact same applicators that you see when you're putting in the ear tags, but in this case, it actually saves the little tissue sample from where that would go into a vial, and you put it right back in to the box and send it in to the companies for genotyping. Other options that you can use are nasal swabs. One of the most common companies that have nasal swabs for livestock is Performagene, or DNA Genotech, and the nasal swab is called Performagene. Really easy, especially if you consider, any animal welfare. Sometimes people frown upon the tissue applicators, but I think for most of us it all works fine. But nasal swabs are an option. They do tend to be a little bit more costly, such as about $5 to $6 per swab. So that's usually the drawback with the nasal swabs. And lastly, we can also get DNA from semen samples. So all of these are possibilities. So next we're going to move on to what actually is included in genetic evaluation. So what do you get back after you submit your sample? So the big thing is most commonly you get these main performance index. And there's a couple different indexes out there, and we're going to go over the three that are the most common. So the first one is called net merit. So this is a primary performance index that's calculated by the CDCD with the research by the USDA. And the big deal here is that it's the same weighting of traits regardless of breed. So you could look at basically the net merit of a Holstein compared to a Jersey, any breed that you were interested in that we have data on. The other unique part about net merit is that it is based on economic values of the traits. And this is stuff that I did put the websites down here, but you're always welcome to go in and look at how do they generate the economic value of, say, calving ability or protein or productive life. So net merit has economic value with each of their traits. What I did, this pie chart, it kind of breaks down each little thing, but big picture is about 45% of the traits are production traits, mostly protein and fat. 41% are considered health traits, and then 16% are conformation traits. So that's kind of the big picture of how net merit is categorized. The next thing about performance index is that you also have these performance indexes that are generated by industry or by breed association. So most commonly for Holstein, you have TPI, Total Performance Index, Jersey, the Jersey Performance Index, and for instance, GenX, one of the companies, has the ICC, which is Ideal Commercial Cows. So those are just some examples. With that, they're generally geared towards breed or company ideals. So I'm going to show you in the next couple slides how these all change just a little bit. So there's mainly a different weighting in those traits of performance, health, and conformation. Some of them are becoming more similar to net merit calculations. And really this is driven upon by producers and the industry. What is most important to that group? And lastly, private industries, when you submit, say, genetic value, if it's given to you by GenX, on bulls, or select sires, they tend to default to certain performance indexes. So GenX often uses lifetime net merit, whereas select sires normally will default for Holsteins to TPI. And internationally, TPI is one of the more well-known performance indexes, particularly because of the influence of Interval. Interval is a larger group of international collaborations that are really trying to come up with these performance indexes that you can compare an animal from the United States to New Zealand to Germany. That's the goal. And right now, for the most part, performance indexes are going to be different for each country, because in our case, if you compared, say, the U.S. to New Zealand, most of our animals and farms are going to be very commercial based, whereas the farms in New Zealand were going to be more pasture based. So they're selecting for a different type of animal. So just to give you some ideas, if you look on the Holstein website, this is the Holstein Performance Index, or TPI, and this shows you the different weighting of traits. Big picture is they also have some unique smaller indexes such as feed efficiency that's included, or fertility index. If you look on the Jersey website, you'll get something like this, a pie graph that shows you what they include in their performance index. The raw data that goes into this is the same. However, how they break it down, here in Jersey, you actually see functional trait index. So So how they break the data down is what's different, not what goes into the original formula. So putting all three of those together, net merit, TPI, and JPI, you can see how there's, both similarities and differences. Production is basically higher. There's a higher weight for production traits in Jersey, whereas opposed to net merit and TPI are pretty similar in production. However, if you compare TPI to either of the other two, they weight conformation traits more heavily. So big picture, if you break all of those components down into a similar categories, this is how they're both similar and different from one another. Under net merit, I have down Holstein, Jersey, Ayrshire, Brown Swiss, Guernsey, and Milking Shorthorn. These are the breeds that the CDCB currently has enough of animals in their database to generate genetic evaluations for. Now what we're moving into is to show you what would the data actually look like for you as the producer when you get this back. So for example, this is a CLARIFIDE report. They come in an Excel format. You can look at them online in their Light program. If you download this, the very first page is basically your table of contents here. And the nice thing is that some of these really are just different ways to sort the same data. So it's a repeat of the same data, and I'll show you a little of that. Other pages are very specific to such as parentage. And then there's also what I call kind of your definitions page, that all of the abbreviations are explained, so what they mean and what the information means. So for instance, the sheet that says core traits, what you're going to get is your animal IDs, the breed IDs, and then you're going to see some of these major indexes here. The net merits, the CDCB will also generate some very specific indexes such as cheese merit, fluid merit, and grazing merits. These are Holsteins that we're evaluating here. So we have TPI, we have some individual scores, and so this is the major core traits page. Similarly, there's a page that's called composite indexes. So you can see some of this data is absolutely the same. It's just repeated and presented in a slightly different fashion. So again, you have those same couple: net merit, cheese merit, fluid and grazing merit, and TPI. But here you also have feed efficiency and fertility index. Those are both generated using the Holstein Breed Association scores. So this is what the composite index page would look like. Another page that probably isn't used as often by producers is the one that's genetic conditions. So these are specific diseases that we know of, that we have a genetic, we can basically look at if your animal is a carrier or if they're free of being a carrier. So this would have in mind what these HH1 through HH5 are. Each of these are Holstein fertility, recessive fertility haplotypes. So an animal that's a carrier, you wouldn't necessarily notice that they have an issue with fertility. However, if you breed them with another carrier, then you're likely to get these early aborted fetus or embryos. So the abortion is so early in the embryo development that often you wouldn't even see the manifestation, other than it would take a lot longer to get this animal bred. But this is something where you could, if you want to look at increasing or improving reproduction in your herd, you could look at this and either look at who are the carriers and who is free, and that might help in culling decisions. Or if you do have a carrier, that doesn't mean you have to call that animal. It might be a great producing animal, but you just want to avoid breeding it to a bull that's a carrier as well. So this is how that page can be used. This information on the genetic conditions is not directly in the genetic evaluation score that you receive. One of the other pages that's in these genetic evaluations is parentage. So as I mentioned, when you put in your submission, you're asked to put in the dam and sire information. The genetic will basically show you this is what you submitted, this is what the genomics find that the parents, the sire and dam are, and does that actually agree? So if there's a disagreement between what you submitted and the genetics, they will basically put that in the report for you. And just to kind of bring it up, one of the benefits of Zoetis, I'm not trying to push one company over the other, but I will say they have a really nice interactive online program. And if you want to kind of see what is the overall progress of your herd, you can kind of compare that to breed standards. So that's one of the benefits of those. Not moving. Hold on just a second. Okay, now we move. Sorry about that. So the next section is the last section. I kind of inundated you with all of the stuff you get back. So how do you actually use that information now? So the first thing to consider is that you have to actually realize and accept, you have to accept that genetics matter to you. So they are not the all-encompassing answer, they're just part of the equation. So genetics provide basically that foundation for potential for your animal. They set a genetic ceiling for the herd. So you can have the best environment, the best management you want, but if you don't provide the genetics for those animals to exceed or reach that potential, that's the genetic ceiling of the herd. Likewise, you can have the best genetic animal, and if you don't provide really good management in the environment to allow it to reach that potential, it's not going to reach its genetic potential. So you need to incorporate all of those factors together. Genetics is just a piece of the pie, but it's an important piece of the pie. With that said, there will be animals that exceed the estimated potential. Likewise, there will be animals that don't achieve the expected potential. Every once in a while, you will have the case of, say, a calf that has a fantastic potential, but it's gotten respiratory disease, 3 times, you're going to call that animal. So that is a fact that's going to happen. It's not the overwhelming fact, but it will happen. The other thing is, even farms that don't use genetic information, they're generally going to still improve due to overall industry improvement. The bottom line is that improvement will be at a much slower rate. They're basically the ones that are following on the tailcoats. So I talked about all of these big performance indexes that are generated. We have net merit, TPI, JPI. They are a great starting point. If you select animals based on these, what you should be doing is improving your animals on production, health, and conformation, all of those things at a single time. But what I would suggest, if you really want to see the benefit and faster improvements, you need to use this as a starting point, not as just the start and end point. So, big picture, establish your herd goals, and for most farms, they've already done this. But with that, identify the methods, in this case, the genetic methods, to achieve those goals. So what that's going to do is it's going to really focus the use of your genetic information, that information that was returned to you, how do you use it? And with that in mind, when you focus that information, you're going to reduce the time that it takes you to achieve that gain. So I saw this before. I like this idea. It's just called smart decision making. So I put an example here. If we want to basically start with specific, our goal is specific. We want to improve reproductive efficiency within the overall herd. So is it meaningful? Of course it is. You're going to reduce your costs that are related to related to reproductive problems. You'll improve both the health and the welfare of your animals. What is the action? How are you going to tackle this? And we're talking about genetic evaluations. So in this case, if I wanted to improve reproductive efficiency, I might start with net merit or TPI or JPI. Start with one of those overall indexes. From that, then you might go a little bit more focused. So what this means is you might initially sort all of your animals and look for, the animals that have really good net merit to the ones that have poor net merit, and you might start with some decisions there. But with that information, the next thing, maybe you're going to start sorting those animals on their fertility index if you're using Holsteins. Fertility index has heifer conception rate, cow conception rate, and daughter pregnancy rate in it, and you can see how they weight those traits. If you don't quite like the weighting of the traits, or maybe you have a Jersey and you're not even looking at Holstein, you can still use individual traits. And maybe you're more concerned about conception rates than calving ease, or maybe you want all of those things. You can factor them in yourselves. And lastly, if we're also thinking about fertility, we can actually look at those genetic conditions, those fertility haplotypes, for instance. So maybe we keep those animals, but we breed wisely. Don't breed two carriers together. So those are all the different things that you can use in your evaluations other than just net merit to actually improve reproductive efficiency. So what's the end point? So with that in mind, who do you genotype? How do you implement this? What's the cost and what's the time frame? The more selective you are in choosing and basically implementing this, the faster you're going to see a gain. And lastly, is it tractable? You know, can you actually look back at your herd from the time of where you started to after you've implemented genetic selection and see that you've made progress? So this brings us to the next phase of who do you actually genotype? So you should start with, I think, a couple questions that you ask yourself. Firstly, what type of traits do you want changed in your herd? Are these traits easily characterized, or are they more difficult and complex traits? So milk yield versus, say, ketosis. Milk yield, you're getting data on that every day on your farm. You can easily characterize it, and you can make your own decisions on, without any genetic information, on who to keep and who to call. Versus ketosis, yes, you can see that, but there's a lot of information out there that says how much subclinical ketosis costs a farm, and that's really hard to characterize. So if you have a genetic marker that's associated with ketosis, that's going to be extremely valuable. A genetic marker that's associated to milk production, yes, it's still valuable. Remember, we can gain faster improvements using the genomic information as well. However, you will still be able to improve just selecting based on the phenotypic information. So this is one thing to consider. Sorry guys, we have a delay. There we go. The other thing is, what's the time frame of change? What do you want to see? You know, do you want to think about that idea where you don't use any genetic information and know because the industry is improving, you will eventually improve as well? That's one option. Another option is the AI companies are very active in genetic selection, and you can make use of that. You know, you can put it out there and be very specific about what bulls you want. That example of reproductive efficiency, so you choose the bulls that have not only reasonable and good net merit, but the ones that are particularly high in their reproductive efficiency. The other thing is herd management. You know, the idea of, are you going to genotype your entire herd versus select groups in your herd? And we'll talk about that in just a minute. And lastly, what's cost effective? You know, how many individuals do you genotype and what platform do you use? So, for instance, if you're looking at using genetic evaluations on bulls, so quite honestly, most of this cost is put forward by the AI companies. So they're the ones paying for raising the bulls, and in most cases, and also for genotyping the animals. Producers will do some of this as well, but a lot of that cost is already put in by the AI companies. So there's less cost to you. On the other hand, you might have to pay higher to use higher quality animals. Marketing. Genetic evaluations are definitely becoming more common to market animals. And then lastly, the most common users of genomic evaluations. The AI companies, through those popular sires, have some of the largest impacts on the population. And I am a huge proponent to put out there that you need to keep in mind the potential for increased inbreeding. When we select on traits, we are selecting, we're generally going to have increased inbreeding because we are selecting individuals that are likely more related, and they're showing these common traits because they're more related. That is also how we often will occasionally get these recessive traits, such as the fertility haplotypes, that will be propagated because of these, the common use of common or popular sires. So for instance, that is how most of the fertility haplotypes can all be traced back to common sires. So just keep that in mind to try to keep some diversity when you're selecting bulls for your herd. So now comes the question that's usually a bit more confusing on how and why or when you should genotype cows, heifers, or calves. So again, the cost, in this case, it's really generally on the producers. AI companies will do some because they are choosing the best cows for siring their bulls or for their select sires. Strategies. So this is a huge debate right now. So you will have some situations where the idea is that you genotype the entire herd. And in some cases, this might work for someone. And there are some of the companies that are really, really pushing this. Biggest issue is the cost. The other options are you can genotype select groups of animals. However, do you genotype, say, just the best animals in your herd, the middle group of animals, or the bottom animals? So some of the debate in that is that sometimes you'll hear people say, well, I would do the middle animals with the reasoning that based on the phenotype, production and health, they can obviously tell the top and the bottom. What they don't know is where the guys in the middle fall. So some people will argue genotype the group in the middle and help use that genetic information to make your decisions then. Other people will say, I'm only going to do the top animals in the herd, because those are the ones that are really the aces in my herd, and so it might help them make fine decisions like, this is the animal I'm going to use for embryo transfer versus this is one that I might be marketing, those types of decisions. And lastly, probably some of the fewest information or debate is over whether you should genotype the bottom group of your herd. And usually this is a situation of, is the bottom group, are you actually replacing animals, or are you keeping all of them? So it has an effect on if you're keeping them, how would you treat them? And if you're calling them, maybe that's not such an issue of you don't need to genotype them in the first place. And then the next thing is the idea of, do you just do calves, or do you wait until they're heifers? So the big part about genotyping calves and heifers is that you're genotyping them early, when they're young, so you get that information, particularly if you're in the idea that some of your management is going to be going into culling decisions, then you can use that genomic information to decide, this is the bottom group. So these are the ones that are going to be culled versus this is, your middle section or the high section of animals. So you can save money, genotype them, make early decisions of who to keep and who to cull in your herd. And then the other thing is, once you've actually genotyped them, you have to use that information. So it's really, how are you going to use that information? So one, if you are culling, that's one very easy way to use that genomic information. Another way is service sires. One of the farms that really implements genomic selection very well and very aggressively tend to basically put their herd, so they started with genotyping just the calves, but now that they've done it for a number of years, basically their entire herd is genotyped. So that's another way where you get to the entire herd, but you don't have to put all of the costs up front at one time. Start with just the calves or heifers, and over a couple years, you will have basically your entire herd genotyped. So what they use is that information to make those decisions on service sire. And they will basically put their group in different rankings and decide, these are the animals for embryo transfer, these are the animals that are going to pay for the highest sires or semen samples, where maybe the next group of animals, they will put reasonable semen into them, and a lower group, they might be the ones that they're going to use lower quality donors. So all of those can play into your decisions, but you definitely have to have a plan of attack. To bring all of this together, one of the main points I wanted to bring back is that genetic evaluations, it's just an estimate of their genetic merit. Traditionally, it was phenotype and pedigree. Now we add the genotype, and it makes it more accurate and faster for selection. So the bottom line is you will more accurately choose the animals to show that gain in your own herd, as well as reduce the time that it takes to get there. There's multiple different estimations of this genetic merit. So there's net merit, TPI, JPI, etc. All of them have their own benefits. You need to decide which one is appropriate for you. Are you looking at production? Are you looking at conformation? Each of them, which one's the best one for you? With that said, it's a good starting point to use those general net merit or performance indexes to start with, but don't forget there's tons of other information in there. When you set particular goals, you can get there a lot faster by using that additional information. So bottom line, identify your herd goals. What's the genetic information to achieve those goals? And then what's your strategy to actually implement that information? What are you going to do with it? So I wanted to bring this up as well because this is always fun, and I like fun stuff. But as I was browsing the internet for genetic pictures of cows, this came up. And it just puts, I mean, it's funny to me, but on the other hand, seriously, it puts a horrible connotation on sometimes when people think of genetics, they really look at it is that we're out there and we're making these Frankenstein animals. And while I personally am a proponent of some of the really scientifically sound genetic modifications, genetic evaluations and genetic selection, you are not altering necessarily anything particular in their genes. We are just selectively breeding. So be very comfortable with that. That if anyone asks you, you are not modifying necessarily anything crazy. We aren't putting, a goat's genes or a bug's genes into your cow. But what you're doing is basically selectively breeding. So it's always just a good reminder. There's different ideas of what people have in mind when you say that you're doing genetic selection. And lastly, I also teach as part of my position here in the animal science department, and I teach the principles of animal genetics, but then I also teach an advanced applied dairy cattle genetics. And last year was the first year I taught this course, and their final project was to put together this website. So this is going to be available probably within December for the public. And really what it is I'm just putting it out there. The students selected their own things that they wanted to put in here. So some of the students were non-dairy students coming into the class. So they might have put more information on what it was like to look at genetic selection from a non-dairy perspective, as well as some of the students were very oriented and came from dairy farms themselves. So they put in more of the ideas of what's the economic impact, what are resources, genetic resources that you can look at. There's also information about different research that's been done. So feel free to look at this, and I'm always interested in feedback to our website. It's really student-driven, but it really is put out there to kind of merge the academics with the producers and even the non-dairy sector of our community. So I'm happy to answer any questions then.
Great job, Heather. Really appreciate that. That was a really nice overview for us. So folks, if you have any questions for Heather, go ahead and type them into the chat box, and we'll see if we can get them answered for you. But while we're waiting for folks to take a minute to type in their questions to the chat box, I wanted to ask a couple of questions of Heather. The first one being is earlier on in your presentation you talked about the different ways to do the DNA sampling. So are the methods that are used, are they dictated by the company that you're sending the sample to, or is this something that a farmer can decide which method they want to use?
In most cases, the farmer can decide what sample they would prefer to use. So if it's one, some places find it very convenient where they are genotyping all the calves. So they're going to go out, they're going to put ID tags in the calves anyways, so you might use the all-flex, and that might just work really well. For the most part, I have never heard of a company that won't take every single one of those types of samples. You do run slight risks of sending in a hair sample. The hair samples, one of their downfalls is you don't get as much DNA or sometimes as high a quality DNA from a hair sample. So every once in a while, if the sample just wasn't enough, they might have to come back and say you need to resubmit the sample. On the other hand, if you did blood, which you get a lot of DNA from, you have to deal with, shipping it on ice. So it's really convenient, but most companies will accept any of those options.
Okay, great, thanks. Thank you for an excellent presentation, Heather. On behalf of PRODAIRY and Dairy Business Communications, I want to thank you very much for this presentation.